Chapter 6, Cell Division. These are the objectives. So in this chapter we are going to focus on cell cycle, and we're going to learn different stages in cell cycle. Then we're going to discuss different types of cell cycle we can see in prokaryotics and eukaryotics. So in prokaryotics, we can see a special type of cell division called binary fission, and we're going to discuss what happens during binary fission. And we're going to also discuss some of terms like chromatids, homologous chromosomes, heterologous chromosomes, and you're going to learn what are the differences between these different terms. And we also are going to discuss mitotic cell division and meiosis cell division. And finally, we're going to discuss how in meiosis different offsprings form. What are the events that happens during meiosis that determine the diversity in offsprings? So as usual, this chapter also has a background story, and this background story comes from a ruin experiment. And this experiment coming from two different labs, and mainly this is some kind of accidental finding. And in one lab they found like a detergent causing abnormal cell growth, and in a different lab they found plastic, a leak in a plastic container causing a difference in their experiment. So first, the scientist group or the lab is Soto and Sonachin. So they were actually studying hormone estrogen and how this hormone affects cell growth. So they were using human breast cancer cell lines. So basically, like these cell lines, they contain only breast cancer cells, and they kind of like you can put it in a small petri dish and in different media and observe how they grow. So here the experiment that they did was they wanted to observe the cell growth in the presence of estrogen and in the absence of estrogen. So normally what happens is like in the absence of estrogen, the cells are not growing. But in this experiment, in the control flask or in the absence of estrogen, the cells starts to grow. So that was the first abnormal finding that they noticed. So the second lab that we're gonna discuss with the abnormal finding was from Patricia Hunt. So she's also a genetist. So she studies genes, and here she was working with mice cells, egg cells, mouse egg cells. So she was looking at specifically the chromosomes in mouse egg cells, and here what she observed was like in the control flask or the control containers, pretty much 40% of the eggs had chromosomal defect. So it kind of like ruined the entire experiment. So in both cases, you're going to learn later. So in this case, like they later found that the plastic container is kind of oozing out a chemical called BPA, and it was acting as estrogen. So that's what caused this abnormal chromosomal growth, or like chromosomal defects in these egg cells. And in the previous case, so it was actually a detergent that they use to wash the plates, or like their growth flowers. So the leftover detergent was causing that abnormal cancer cell growth in the control flowers. So in this case, like both two different chemicals were able to alter the cell division or the cell growth. So here, as I mentioned, both Soto and Hunt lab, they identified toxic chemicals in our environment can cause disrupt in the cell cycle, or they can cause kind of abnormal like cell division or cell growth. So now we have been talking about cell cycle. Let's look at what is the formal definition of the cell cycle. So the definition of the cell cycle is a sequence of events that makes up the life of a typical eukaryotic cell from its origin to the time it divides to produce two daughter cells. So pretty much like the life cycle of a cell. So since it begins its life until it divides to form like a kind of daughter cells. So that's the cell cycle. So there are two stages in the cell cycle, eukaryotic cell cycle: interphase and in the cell division. So most of the cells in our body or in any eukaryote are in interphase. So pretty much 90% of the cells, they are in interphase. So when the cell is in interphase, is doing what it is supposed to do. For example, like if a skin cell is in interphase, so it is doing what it is supposed to do. If kidney cell is in interphase, so it is doing what it is supposed to do. If a heart cell is in interphase, it is doing what it is supposed to do. So pretty much like cell is doing what is function when it is in interphase. So the cell interphase can be subdivided into three different phases: G1, S, G2. And the next one is cell division. So first let's look at interphase. So G1, S, G2. So G stands for gap or growth phases. Okay. So in both G1 and G2, the cell kind of grows. Okay. So in S phase, S stands for synthesis. So during S phase, the DNA is getting synthesized, or the whatever the DNA in the cell, it will get replicated, or the DNA will get doubled. Okay. So now let's look at this cell cycle cartoon. So right here, this is the start. Okay. So here the newly cell starting. So this is kind of G1 phase, cells doing whatever the function is supposed to do. And then G1 is kind of like growing. Okay, so it's increasing the amount of material inside the cell. S, synthesis, so the DNA is getting replicated or it's getting doubled. And G2 again is getting ready for the cell division. So making sure all the materials are doubled within the cell before it enters the cell division phase. Okay. Alright, so as I mentioned, G1, G2, they stand for gap or growth stages, and S is for synthesis. And so in this cell cycle, there are different checkpoints. So pretty much like these checkpoints make sure whatever the steps that is supposed to happen in each stage has successfully happened during each phase. Okay, so for example, like from G1, when it goes to cell move from G1 phase, all the cell material has been divided, like kind of mitochondria, Golgi apparatus, they must be kind of duplicated. Okay, and at the end of the S phase, all the DNA or like chromosomes should be duplicated. Okay, and at the end of the G2, the cell is ready to be entered to the mitosis. So pretty much everything in the cell should be doubled. Okay, so all the cellular organelles and the genetic material should be doubled, and then the spindle that we're going to learn in a later, it should start forming at the end of the G2 phase. So these checkpoints at different stages, they make sure the steps are correctly happening throughout this cell cycle. So we discussed the 3 different phases in interphase. So if you look at the cell division, so this is the cell division state. There are 2 steps, mitosis or cytokinesis. So in the mitosis, the nuclear or genetic material will get divided, and in the cytokinesis, the cell cytoplasm will get divided into 2 cells. So here I didn't talk about what is this G0 phase. So G0 is a kind of like a neutral phase. So some cells, when after they divide, they kind of exit this cell cycle and they kind of like remain in a resting stage or non-dividing stage. So those cells, we call them, they are in a G0 phase. So it's kind of like a resting stage, and they are kind of like not in a cell cycle. So now the question is why the cell division is important. So like when we look at the multicellular organism, we know that with time the cells, some cells get damaged, and then some cells die with time. So in order to replace those damaged and dead cells, the body needs to produce new cells. So that's one of the purpose of cell division, in order to replace the damaged and dead cells. And another purpose is to reproduce. So in order to like continue a species, we need to produce eggs and sperms. So the eggs and sperms are created through cell division. So we will discuss that in later in this lecture. So now before we move on to eukaryotic cell division, let's talk about the basic or primitive cell division that happens in prokaryotes or like bacteria. So if you remember, prokaryotes means they don't have real nuclei. So that means their genetic material is floating inside the cytoplasm. So here this is a bacterial cell. So they have a circular DNA molecule, cytoplasm, plasma membrane, and a cell wall. So in these prokaryotes, the cell division happens through a process called asexual reproduction or binary fission. So in this process, what happens is this circular DNA will replicate by itself, producing two copies of the same genetic material, and then this single cell, it will kind of protrude the cell membrane into the cytoplasm, and then it will create a cell wall in the middle of the cell, creating two cells. Okay, so now the cells will separate, creating two daughter cells. So this process we call binary fission. So this happens only in prokaryotes. So eukaryotic cell division is much more complex, and we know that in eukaryotes the nuclei is there is a true nuclei, and the true nuclei means the genetic material is surrounded by a plasma like nuclear membrane, and the genetic material is in the form of genes, and the genes are located in distinct linear chromosomes. So the chromosomes are kind of like very long threads, and these chromosomes are DNA wrapped around proteins. Okay, so these materials, they kind of like stored inside the nuclei. So in humans, we have 23 pairs of chromosomes. So that would be total of 46 chromosomes. So here, like what happens is like in eukaryotic cells, there are two types of cell division: asexual and sexual reproduction. So asexual reproduction through mitosis. So it happens in some organisms, and in this process, two identical daughter cells will produce with the paired chromosomes or diploid cells. So in the sexual reproduction, what happens is like with the single cell, sperm or eggs will produce, and these are haploid cells. Okay, so for example, in in a human, so in if a cell reproduce asexually, so we have 43 total chromosomes. So asexual reproduction will produce two cells that has 43 chromosomes in each. But in sexual reproduction, what happens is only you're going to have half of the amount of chromosomes per the daughter cells. So the mitotic division, it only happens in eukaryotes. So it generates two identical daughter cells from a single parent cells, occurs after the interphase, and as I mentioned, it has two steps: mitosis and cytokinesis. And next we will look at what are the different stages in mitokinesis. So the first stage in mitosis and cytokinesis. So the mitosis is the division of the genetic material. Cytokinesis is the division of cytoplasm or the cell. So next, let's look at what are the different stages in mitosis. So these are the main four stages in mitosis: prophase, metaphase, anaphase, and telophase. So these four phases are divided depending on what happened to the chromosomes inside the cell. Okay, so during the prophase, the chromosomes, they kind of become sausage-like structures. They become condensed. So normally in like a resting cell, the chromosomes are long thread-like structures. But when it is preparing to divide, the chromosomes become condensed and short. So they become thick and condensed. So they are pretty much like sausages. So in the prophase, the chromosomes become condensed structures. And during the metaphase, these condensed chromosomes, they align on the midline of the middle of the cell. Okay. In the anaphase, and the chromosomes, so in the metaphase, so all the chromosomes that align in the middle of the cells, they are attached to the spindle. Okay, so I will show you in the next slide. So in the anaphase, what happens is those chromosomes will pull towards the opposite side of the cell. And the telophase, and the chromosomes will uncondensed, and then the nuclear membrane will start to reform. So now let's look at these images. So here this is the interphase. So these are the two prophase and metaphase. So as I mentioned, so here these are uncondensed chromosomes. You can see these are long thread-like structures. Okay, but when it comes to prophase, they become visible. Okay, so they become these condensed short structures. Okay, so they become condensed, and so the reason is like during cell division they move a lot. So if they are like thread-like structures, there is a higher chance they will get tangled and broken into pieces. But when it becomes condensed, it is easier to move around, and there is a less chance of getting broken apart. Okay. So now during the metaphase, all these, so in this picture, these blue color regions, these are the condensed chromosomes, and these red color lines, that's the spindle formation. Okay. And now you can see in the metaphase, all these chromosomes, they are aligned in the middle of the cell, and the spindle is attached to the center of the chromosome, or the centromere. Okay. So now during anaphase, what happens is these chromosomes, so these single threads we call chromatids, and they're gonna pulled into the opposite side of the cell, so opposite poles of the cell by these spindle threads. So in the telophase, these chromosomes, they will start to uncondense or decondensing, and then the spindle will disappear, and the nuclear membrane will start to reform. So now in the cytokinesis, I mentioned that is the division of the cytoplasm. So this in the middle, like a furrow, or the plasma membrane will kind of merge into the cell, dividing the cell into two cells. So here I mentioned that I talked about chromosomes and sister chromatids. Okay, so these are the differences. Okay, so here this is a single chromosome during the gap phase. So during S phase, these chromosomes will get replicated, or it will get doubled. So this is the centromere, where the chromosome is starting to double, and now this long thread become to two threads. So this single thread we call a chromatid. So now this is like kind of like a single unit of chromosome. It become doubled at the end of S phase. So now at the end of the prophase, this is how that chromosome look like. So these are like the sister chromatids that kind of replicated at the end of at S phase. Okay, so when these two separated, each cell is going to get a copy of this chromosome. So as I mentioned, so there are checkpoints in this cell cycle. So these are the main three checkpoints: G1 to S checkpoint, G2 to M checkpoints, and spindle checkpoint. So G1 to S checkpoint, so this at this checkpoint the cell makes sure the cell has enough nutrients, or the cell has properly doubled its organelles. Okay, and now the genetic material will get doubled at the G2M checkpoint. So the cell cycle kind of like, again the cell checks whether it has enough nutrients to go on, and then it also check whether any of the DNA is damaged, or whether any DNA replication is incomplete. So if the cell find out if any of these things happen, whether a DNA is damaged or DNA replication has incomplete incomplete DNA replication, then what happens? The cell will stop the cell cycle, and then it will correct that mistake before it move on to the next cell cycle, next step in the cell cycle. So the third checkpoint is spindle checkpoint. So in this checkpoint, the cell makes sure all the chromosomes are properly attached to the spindle. And if any of the chromosomes are not properly attached, then the cell will not move to the next step unless move to the next step, and then it will make sure it's properly attached. Okay, it will correct its mistake before it move to the next step. So sometimes the cancer happens when the cells have uncontrolled cell division. Okay, so we call a tumor a cell mass created by rapid runaway cell division. Okay, and we call a benign tumor when this uncontrolled cell division is confined to one side. So normally if it is a benign tumor, it can be surgically removed, and normally it's not a threat to the patient's survival. And so these benign ones, they kind of attached into some place, and then stop dividing if they are detached. Okay, so in order to divide, they must attach to a place. So this kind of phenomena, we call it anchorage dependence of these tumor cells. So the angiogenesis, we call the formation of new blood vessels to the tumor cells or the cancer cells. And if there is formation of blood cells, that means then these cells, they have the transportation. They can kind of enter into the blood system, and they can kind of go travel throughout the body, and then kind of anchorage into different parts of the body, and spread throughout the body. So that's what we call metastasis, the spread of a disease from one organ to another. So in this at the beginning of the chapter, I mentioned the two labs had some issues with their experiment, and the Hunt lab who were working with mouse egg cells, they found out like the containers that they were using is leaking a chemical called BPA, bisphenol A. And because of this chemical, it kind of like mimics estrogen. So that's why even the control containers had chromosomal disorders or genetic deformities in their chromosomes. So now, like we know that this BPA is toxic for human health, and nowadays, like when we made these plastic bottles or any water bottles that we use, the industry is trying to get rid of prepare without BPA. So now let's look at the next type of cell division called meiosis. So meiosis happens specifically during sexual reproduction. So there is sexual reproduction, production of the gametes. So the gametes are haploid. So if there is a 2N cell, so that means 2 sets of chromosomes in the parent cell. Haploid means it only get 1 set of chromosomes or N number of chromosomes. Okay. So we call this 2N means diploid, single N means haploid. So as I mentioned earlier, in mitotic cell, in all the somatic cells, the mitosis happens. In reproductive cell, meiosis happens. So now, before we go on to the meiosis, let's look what happens during the fertilization. So in female and male, so the mother has 2N diploid egg cells. Father has 2N diploid sperm cells. So when these two cells go through meiosis, egg cell produce egg and the sperm cell produces sperm that are haploid. So egg is haploid and the sperm is haploid, N and N. So when these two get fertilized, then the baby has 2N diploid zygote. Okay, so in that way, all the human species will continue to have 2N or diploid chromosomes. So imagine if there is no meiosis. So then like 2N and 2N, it will become 4N. And then that 4N and 4N become 8N. So it will be like completely different organism. Okay. So in this way, the species is maintained as it is. So now, when we look at the meiosis has two parts, meiosis 1 and meiosis 2. So meiosis 2 is very similar to mitosis. Okay, so let's look at meiosis 1. Okay, so in meiosis 1, what happen is like in the number of chromosomes will divided half and half. Okay, so here it started with the diploid cell and now it end with the 2 haploid cells. Okay, so here you can see these are blue and red. So these are chromosomes and these are homologous chromosomes. So I mentioned that we have 23 pairs of chromosomes. So that means like each pair, so number 1 chromosome, number 2 chromosomes, they are called homologous. The reason is they have similar genes in both copies of these chromosomes. Okay, and they are very similar and like they have very minor changes in these 2 chromosomes. Okay, so again prophase 1, metaphase 1, anaphase 1, telophase and cytokinesis 1. So during prophase 1, the chromosomes become condensed. During metaphase 1, the homologous chromosomes will align in the middle of the cell. During anaphase 1, the homologous chromosomes will separate or pull towards the opposite direction of the cell. And telophase and cytokinesis 1, the homologous chromosomes, they will start to decondense, and then the nuclear membrane will form. Okay, so that's the end of the meiosis 1. So meiosis 2, again there are 4 stages. So this is very similar to mitosis cell division. Okay, so prophase 2, metaphase 2, anaphase 2, and telophase 2 and cytokinesis. So now these are like condensed chromosomes. Okay, so what happens is like prophase, they are already condensed. Metaphase, the chromosomes will align in the middle of the cell. And then anaphase, the spindle will pull these sister chromatids towards the opposite direction of the cell. And now these separated sister chromatids will wrapped around by a nuclear membrane to form the new nuclei, and at the end of the cytokinesis, the cell cytoplasm will divide and create in 2 new cells. So that's what we discussed, meiosis 1. So meiosis 1, 2 homologous chromosomes pairs are separate into 2 different daughter cells. Okay, and here it produces 2 haploid cells. So meiosis 2 is pretty similar to the mitosis. So at the end of this, there are 4 haploid cells. So this is also like from from the background story. So Hunt discovered that BPA hinders the correct separation of the chromosomes into 4 haploid cells during the meiosis. Okay, so pretty much like this is the direct effect of BPA. So it kind of affects the chromosomal segregation during the meiosis. So now, when you look at the meiosis process, like all the 4 cells, they are pretty similar. But if you look at even the offsprings in the same family, they are very different. They are similar to the parents, but they do have some differences too. So these differences, the genetic diversity, is coming from some of the mechanisms that happens in the meiosis process. So these mechanisms are called crossing over. So that's one mechanism. And the other mechanism is called independent assortment of the paired up chromosomes. Okay. So first let's look at what is the crossing over. Okay. So crossing over is an event of mixing up DNA in the sister like 2 homologous chromosomes. Okay, so here these are the homologous chromosomes, blue and red. Okay, so at this point they are not mixed, but when they are closer to each other, these chromosomes, their arms, they can overlap on each other and exchange this genetic material. Okay, so when this at this exchange point, these 2 can exchange. Part of the blue can go into the red chromosome, and part of the red chromosome can get exchanged into the blue chromosome. So this is how it looks like at the end of the crossing over. Okay, so now in this case, the different eggs, these egg cells or sperm cells, they're going to have differences in their genes because of this crossing over event. And then the independent assortment. So this is the second event that caused the genetic diversity. So in this one, what happened is these two chromosomes, they can have the opportunity to go into any of the combinations. Okay, so this particular sister chromatids can pair up with this chromatid. Okay, so this one can pair up with this chromatid. So there are so many combinations that pairing up with these maternal homolog and paternal chromosomes in these when they make the gametes. So that's what we call independent assortment that causing the genetic diversity. So now, so the take-home message is like in this study, like we found the chemicals can change the cell's behavior. So now with the current research and current knowledge, so we know that like some chemicals like BPA can cause various diseases like breast and prostate cancer, miscarriages, birth defects, diabetes, obesity, and even ADHD. So a BPA has been found in human blood, urine, breast milk, and amniotic fluid. So it can get into our body. So BPA is banned from baby bottles and children's drinking cup, but still allowed in other types of containers. So always like this kind of research, they bring something valuable to the society. So the take home message is like you have to be aware of what you are using, and any chemical can have like side effect. But with this study we know that the chemicals can affect the cell division and cell growth. Thank you.